This is Soumyadipta Shah, student of sixth semester from the Department of Molecular Biology Biotechnology, Cotton University. I am about to present this dissertation topic, a comparative study of the efficiency of household disinfectants on microbial growth. To begin with the introduction, in hospitals and other healthcare facilities, antiseptics and disinfectants are frequently used on various topical and hard surface applications. Proper sterilization and disinfection can reduce the danger of pathogenic transmission and cross contamination. Because in hospitals, there is a term called hospital induced infections, which is one of the major causes of death annually. And healthy people catch different infections in hospital while they are being treated for other kind of diseases, so it is one of the major causes of that. So this has to be taken care of. So and various disinfection and sterilization techniques are used in hospitals and to kill the microbes so that infections can be reduced. Methylated spirits, which are primarily alcohol, significantly reduce skin flora and can be used and are also used for cleaning the surfaces in the hospitals, and also for screen preparation prior to injection or surgical preparation, making them generally acceptable disinfectants. It has been demonstrated that they have a cleansing effect on burns and wounds also. Before grafting or patching any kind of burns or wounds, they are properly cleaned so that there is no not presence of any kind of microbes which might cause infections at a later stage. And also, disinfection should only be used when there is no other option available of sterilization, uh, because it is sterilization is always better than disinfection. A disinfectant is any substance intended to destroy, kill, prevent, or slow down the reproduction or the growth of bacteria. Disinfectants work by destroying the protective membrane or protein envelope that covers a microbe. There are several different types of pathways by which a disinfectant can kill the microbe. The first one will be solubilizing cell components. The second one will be interfere with the molecule of transport system. The third one will be denature of protein, and the fourth one will be degradation of the biological functions. Now, this is a review of literature which I have collected by going through the research papers present and the review articles present all over the web. So, to begin with, disinfectants are chemical compounds used to kill microorganisms in water treatment plants, public sanitations. Not only this, hospitals and also in households. They can have both antiseptic and disinfectant properties depending upon their concentration. For example, phenol at 0.2% concentration behaves as an antiseptic, so which is harmless to the tissue, but at 0. So at 1% or concentration, it behaves as disinfectants, which is harmful to the tissue. This mechanism of action involves disrupting cell membranes, leading to cell death, and its effectiveness depends upon the concentration used. There are seven main disinfectants which include alcohols, hypochlorites, iodophores, phenols, phenol disinfectants, and quaternary ammonium compounds. Formaldehyde and butyraldehyde are fast-acting disinfectants used in hospitals for surgical instruments, hypochlorite for water and sewage systems, and phenol for large surfaces. Bacteria's membrane protect them from the environment, and the bacterial cell wall provides rigidity which varies between gram positive and gram negative bacteria. Nutrients enter the plasma membrane through passive diffusion and active transport. Cross infection, as I have already discussed, is one of the global concerns, and it causes a lot of death. Hospital induced infections kills more people than any other disease causes, because it is a hidden infection, and most of the people are not even aware of this kind of infections. Whenever they go to hospitals to get treated for any kind of other disease, they catch this infections, which cause a lot of problems and is one of the major killers. Dry heat, unsaturated chemical vapor sterilization, and chemical surface disinfectants are low cost, time efficient method to combat these issues. Now, the major objectives of this experiment of the dissertation work is that to examine using the agar-based diffusion method effectiveness of various widely used household disinfectants against bacteria, to ascertain the resistance of the test bacteria in this study to various antibiotics, including chloramphenicol, tetracycline, and gentamicin, to conduct biochemical tests to characterize and identify the test bacteria used in this study. So first of all, we started with the collection of sample. We collected the sample and then we put this bacteria in in a solution of a nutrient broth where we allowed the bacteria to grow for 24 hours. Then we checked, and and again we kept it for another 24 hours, so in total we kept it for 48 hours, and then with which was kept at 37 degrees celsius and with constant shaking at 120 rpm. And then there was bacterial growth. Then we took that bacterial strain, and then we put the bacteria into nutrient agar plates, and then the bacteria was identified and classified. Now from those bacterial growth plates, we patch bacterial colonies, and then after growing those patch colonies, we then made pure cultures from but a particular patch colony, so that we get a uniform bacterial strain, because one colony consists of specifically most probably contains only a single strain of bacteria. So even if there is any kind of discrepancies which might come up in our experiment, and we have to go back few steps back, so we can always come back to this particular patched plate, and we can directly get the same strain of bacteria to repeat our experiments all over again. And later on, with the lawns that we have got, we use the Kirby-Boer method, or the Dix diffusion method, by which we calculated the diameter of zone of inhibition, and on the basis of for different constituents and concentration of disinfectants, and later on we came to a conclusion which we will discuss later. So this is the schematic diagram of what we have done. First of all, we have collected with sterile swabs. Then we have kept the bacteria in nutrient broth. Then we have prepared nutrient agar plates and then grown the bacteria. Then we patched the nutrient agar plates, and then we ultimately produced uniform lawns, and then we have done the Dix diffusion method to analyze and differentiate and classify and differentiate and compare the zone of inhibition of different concentration of disinfectants and antibiotics. So this is the workflow that I have already said in the previous slide. Now we have used three different types of disinfectants at various concentrations of 50%, 75%, and 100% concentration. So the first is that we have used is Lysol. The second we have used is Savlon, and the third one that we have used is Dettol. Now, while doing this experiment, we had to prepare the loaded disinfectant disc. So for that, we used Whitman 3 filter paper, and using a stencil, we had cut 5 mm diameter small discs, and then we have soaked those discs into the specific concentration of the disinfectants, and then we have kept it to dry in hot air oven for 30 minutes. Then we have used the sorry the discs were then autoclaved and soaked in the triplicate concentration of each sample and dried in a hot air oven, maintaining sterility for 30 minutes. And then we have calculated by using the measuring scale. Now, after 48 hours of incubation of Bacillus subtilis at 37 degrees Celsius, we have got 9 different readings, and based upon that, we have classified the findings into this graph. The x-axis denotes the disinfectants at different concentrations, and the y-axis denotes the extent of inhibition in a millimeter, which is the diameter essentially. Now, for Lysol, we will see that the zone of inhibition is 25 mm at 50 degree, 50% concentration, and 21 millimeters at 75 degree of 75% of concentration. And at 100% concentration, the zone of inhibition is reduced. Similarly, Savlon has the highest zone of inhibition at 75% concentration, and Dettol has the highest at 100% concentration. For Escherichia coli at 37 degrees Celsius after 48 hours of incubation, we have again taken the 9 different types of readings, and we have got a different type of analogy. For Lysol, the highest zone of inhibition was seen at 50% concentration. For Savlon, the highest was again seen at 75% concentration, and for Dettol, it was seen at 50% of the concentration. Now, looking at this graph, we can see that the Bacillus subtilis was more inhibited than E. coli. So we can draw a comparative study from here that the disinfectants were working properly on gram positive bacteria, and were working less effectively at various concentrations at on gram negative bacteria, that is E. coli. Now, from this we can say that from this diagram, we can also come to the conclusion that Lysol works best at 50% volume by volume in both the Bacillus subtilis and E. coli, and as the disinfectant concentration is raised to about 100% volume by volume, the extent of inhibition decreased by 5% and 33% respectively. Savlon, that we have seen earlier, works best at 75% volume by volume in both Bacillus subtilis, that is a gram positive bacteria, and E. coli, that is a gram negative bacteria. And as the concentration is decreased to further 50% concentration, we see that there is further decrease of by 5.6 and 50% respectively. As the concentration of Dettol increases, the extent of inhibition increases by 50%. And also there is a change in the concentration of Dettol. There is a change in the inhibition of Dettol when we increase the concentration to 75%, and from 14.3% to 100%. Thus, Dettol is better disinfectant from gram positive for gram positive bacteria than gram negative bacteria. This is the conclusion that we can draw. Now, moving on to the next thing that we have done to compare the effect of the effect of disc and the in the experiment, we have directly used droplets without putting them in discs, and again done the same process. But here we have seen that there is some kind of difference in the readings and zone of inhibition when we have used direct droplets. Maybe the discs that we used did not allow more disinfectants to spread, and might have absorbed some of it and did not allow to spread it when it was put in the bacterial lawn. And also we have done the antibiotic test on bacterial lawn, and we have got different readings, and we have compared the zone of inhibitions between different disinfectants in both the gram positive and gram negative bacteria, that is Bacillus subtilis and Escherichia coli respectively. So from this given data, we come to the conclusion that gentamicin has the highest zone of inhibition both in Bacillus subtilis and E. coli partially, but E. coli, the zone of inhibition in E. coli was more in tetracycline, E. coli being a gram negative bacteria. So for a gram positive bacteria, the highest inhibition was shown by gentamicin, and for gram negative bacteria, the highest inhibition was shown by tetracycline. Now moving on to the next inference from the previous experiment, we have seen that gentamicin is most effective antibiotic for inhibiting gram-positive bacteria and gram-negative bacteria, while chloramphenicol shows minimal inhibition. Savlon is a good disinfectant, but poor results may be due to a heat-level chemical component inactivated during the experiment. This conformational change may decrease reaction efficiency and stop the growth of bacteria. As we have seen that Savlon worked best at 75% concentration in both gram-positive and gram-negative bacteria, but when we had heated the Savlon solution, it reduced considerably. So that might be one of the reasons. But here again, from this particular graph, we can come to a conclusion that gram-positive bacteria are more properly inhibited than gram-negative bacteria. And also from this graph, we can see that the green color graph which shows the rate of zone of inhibition of Bacillus subtilis, that is a gram-positive bacteria, to E. coli, which is a gram-negative bacteria. Gram-positive bacteria here again holds a much larger area of inhibition, whereas gram-negative bacteria, that is E. coli, does not have that much area of inhibition as compared to Bacillus subtilis. Now, the the next experiment that we did was swab sample from laboratory. As we have earlier collected samples from various sources, this experiment was done to see the type of inhibition present in the locally acquired bacteria from the laboratory. And we have done it in a similar way. We have used Lysol, Savlon, and Dettol at various concentrations. We had make a triplicate for 100, 75, and 50% concentration of each disinfectant, and also we have used three different antibiotic discs: chloramphenicol, gentamicin, and tetracycline. And this culture was grown overnight using swab samples that was taken from the lab's hotspot areas during the final lab of the experiment, as I have already said. In a similar way, we turned the sample into a lawn. By calculating the zone of inhibition after 48 hours of incubation in the culture, we tested for resistance using the same detergents and antibiotics. And we had come to the conclusion that the zone of inhibition is maximum for pure concentration, and all the three disinfectants almost show similar zones of inhibition at different concentrations. This is the inference from the data that we have acquired after doing the swab sample, and also comparing with the previous experimental data. But we have come to another conclusion that Lysol and Dettol coincide on their zones of inhibition, where Savlon at all the three different concentrations show more zone of inhibition than the other two. So this is one of the inferences that we have drawn. Now this is the result that we have come across after all the considerations and the experiments that we have done. Savlon zone of inhibition at 100% of the concentration exhibited maximum zone of inhibition, followed by Savlon at 50% and 75%. Overall, we can infer that the most recent Savlon is better disinfectant for broad spectrum uses in both homes and laboratories. It is important to note that the effectiveness of a disinfectant can vary depending on the type of microorganism it is being used upon. It is also worth considering the factors such as cost, availability, and environmental impact when selecting a disinfectant for use. What kind of bacteria on which we are using the disinfectant also plays a very important role. Not only bacteria, the other kind of microbes like viruses and fungi can also be taken under consideration. Overall, through our experiment, we have come to the conclusion that Savlon may be a good choice for many applications, but other considerations has to be also taken, like the different type of organisms that we are using. Now, the to the end part of this presentation, the discussion, environmental microorganisms are causing infections and bacterial transmission from hand to food objects or people in a significant factor. Proper hand hygiene practices such as washing hands with soap and water can reduce the risk of spreading pathogens. Alcohol-based detergents like Dettol, Savlon, and Lysol are effective against bacteria, viruses, and fungi. The right type of the follow manufacturer, the right type and the best way is to follow the manufacturer's instruction for maximum effectiveness. But however, we have seen that the manufacturer's effectiveness is not what they claim exactly. Most of the disinfectants and the sanitizer companies or the products, they claim to have almost 99.9% efficiency, which is not the case. Maybe the efficiency reduced to 50% or 60%, but it is never 99.9%. So that is a misnomer, or we can say that that is one of the advertising red flags in the industry. And to the end part of this presentation, the conclusion, the the potency of disinfectants is critical for improving the efficiency in reducing microbial population, which include disease transmission and infection prevention. The antimicrobial efficacy of disinfectants must be determined in order to accomplish comprehensive pathogenic disinfection. The disinfectants appear to a broad spectrum of activity, including activity against both gram-positive and gram-negative bacteria. As in our study, we have seen the gram-positive bacteria which we had taken as Bacillus subtilis, and the gram-negative bacteria that we had taken at Escherichia coli. As a result, disinfectants for widespread use should be able to kill a wide variety of common or prospective pathogens. So a good disinfectant is that disinfectants which work at optimum concentration and is a broad spectrum disinfectant, and can kill large or can reduce the growth or inhibit the growth of large number of organisms, irrespective of what type of organisms they are, being gram positive, gram negative, a fungi or a virus. It should be able to stop the growth or the stop the growth of those microorganisms, and not to be limited to a particular kind of organism. And it should be cost effective, and it should be potent. So this was all about my presentation about my topic. Thank you.